how genes regulate the enzyme synthesis. So that concept here we will clarify. We know gene is unit of heredity. Genes synthesize specific enzyme. That enzyme may act as a protein or may be useful for carrying out a particular reaction. And the foreign proteins, they controls specific trait of living organism or specific character of living organism. Total amount of DNA present in cell may contain few to thousands of genes. One should know that different enzymes are synthesized in the cells in different environmental conditions and at different stages of growth, and also at the time of differentiation of an organism. It means the necessary of expression of gene is not at one time, because in different stages of growth different types of enzymes are required for the metabolism of different substrates. It is essential to regulate the activity of the genes. Even in case of unicellular organisms like bacteria, cell does not produce all the enzymes all the time. All genes are not active at one time. Certain genes become active and functions for synthesizing the enzyme, while other genes become inactive and they do not synthesize enzymes. Genes which are functioning for the enzyme synthesis are said to be switched on, and the genes that are not functioning for the synthesis, call them as switched off. Means genes are switched on and switched off at different times in the life of the organism. Thus, genes have control mechanism for synthesis, enzyme synthesis. Gene regulation in case of prokaryotes, how it happens, that we will see. And what is inducible enzymes, that we will try to understand. Take an example of prokaryotic cell, for example, bacterial cell, and particularly Escherichia coli. So this E. coli is capable of producing many enzymes, but only necessary enzymes are synthesized during particular time. The enzymes are synthesized in required amount by regulatory mechanism of gene action. Otherwise, this will not happen. If there is no regulatory mechanism, then this Escherichia coli cell might have flooded with the enzymes, as all of the genes might have become switched on or functional, and they would have produced enzymes, and entire cell might become flooded with the enzymes, and this is unnecessary, and that's why there must be a regulatory mechanism, and that regulatory mechanism, how it is there in the cells, living cells, how it operates and all that we are going to study here. So in prokaryotic cells, for example, we have taken Escherichia coli. So such cell is capable of producing many enzymes, but only necessary or required enzymes are produced during a particular time. There is control mechanism that enzymes are synthesized in required amount in this bacterial cell by regulation of gene action. There is category of enzymes in the cells known as inducible enzymes. Sometimes inducible enzyme synthesis can be induced in the cells by substrates, and those substrates are called as inducers. Inducible enzyme synthesis is induced in the cell by some substrates, and if this thing happen, call such substrates as inducers. The process in which inducible enzyme synthesis is induced by inducer. The process in which inducible enzyme synthesis is induced by inducer is called as a induction. But sometimes metabolic end product synthesized may inhibit or repress the synthesis of enzymes. See here, sometime what happens, the end product of particular metabolism may inhibit the synthesis of enzymes, and it is known as a feedback or end product inhibition due to formation of end product. Feedback or end product inhibition, it is called as the substance which represses the synthesis of enzyme, call it as a repressor, and the process is called as a repression. So we have seen here what is inducer, what is induction, what is feedback or end product inhibition, and then what is the repressor and the repression. The inducible enzyme, this set of enzymes, so these enzymes are studied in Escherichia coli. A wild type of bacterium, Escherichia coli, was cultured or grown in laboratory on a medium containing glucose. E. coli was grown in laboratory on culture containing glucose. In such situation, this Escherichia coli cell was able to produce only minute quantity of enzyme named as a beta galactosidase. See here, when grown on glucose containing medium, E. coli was able to synthesize in very minute quantity the enzyme named as a beta galactosidase. when the same wild type of E. coli cells were grown on culture medium containing lactose. Now in culture medium there is lactose. Within few minutes, cells start synthesizing beta-galactosidase. Synthesis of this enzyme, beta-galactosidase, continues until very large amount of beta-galactosidase have been produced. It is obvious that the lactose here, what happens? Lactose induces synthesis of beta-galactosidase in E. coli. So when there was glucose in the medium, beta-galactosidase synthesis was very less, and when such cells were grown in the medium containing lactose, the amount of beta-galactosidase enzyme synthesis was more. The beta-galactosidase is an important enzyme as it breaks down lactose into glucose and galactose. This peculiarity of this enzyme, beta-galactosidase, can break or is able to break lactose into glucose and galactose molecules. And this breakdown is essential because E. coli cells cannot utilize lactose directly, either as carbon or energy source, unless lactose is broken down into simple sugars like glucose and galactose. And this ability of breakdown is to this enzyme beta-galactosidase. So along with beta-galactosidase, in these Escherichia coli cells, there was a report that another two enzymes named as beta-galactoside permease and beta-galactoside transacetylase, they were also produced. So in all, three enzymes, beta-galactosidase, beta-galactoside permease and beta-galactoside transacetylase, these three enzymes were produced in the Escherichia coli cell. These three enzymes, collectively they are called as lac enzymes. Lactose, lac for lactose, lac enzymes. Beta-galactoside permease facilitates the entry of lactose into bacterial cells. The work of beta-galactoside permease to facilitate the entry of lactose into the bacterial cells. Beta-galactoside transacetylase, what is the work of this enzyme is not yet known, but it is produced in the cells. Thus, presence of inducer. So which substrate induces the formation of beta-galactosidase and then beta-galactoside permease and also beta-galactoside transacetylase, that is lactose. When E. coli cells grown in lactose containing medium, then only beta-galactosidase synthesis in sufficient amount was seen. So this inducer, that is lactose in the medium, induces the cells to form the inducible enzymes, all three enzymes. Therefore, these three enzymes are called as inducible enzymes.